Welcome back. Let's continue with our next topic, which is action potential. So what is action potential? Well, action potential, if we look at the definition, this is quite a comprehensive definition. It's a little large, but it tells you a lot about action potential, and I believe this is the best definition that you can think of and write about in the exam. So it's a series of rapidly occurring electrochemical events, which is depolarization and repolarization. It takes place at the membranes of excitable cells. When once initiated, it is propagated along the entire membrane in undiminished amplitude. That is to say, it follows all or none law. Now, I know that's a little too much to take in the first shot, but let's go over these words one by one. A series, meaning there's a step-by-step process taking place over here of different events. Okay, so these are electrochemical. So there's some charge that will change and some chemicals, meaning some ions, are going to move in and out of the cell, and this will cause depolarization, and it'll be followed by repolarization of the cell. And this depolarization means that the inside of the cell is going to become a little more positive, and repolarization means the inside of the cell is going to be a little more negative at the membrane. So this all happens at the membranes, and it happens in excitable cells. Now let's just talk about excitable cells for a moment. When we talk about excitable cells, we mean cells that communicate and transmit messages using electrical changes in the membrane, meaning action potential. And there are only a few tissues in the body that can give you this kind of an action potential. One of them is the neuron, which we will be studying now. So neuron, then you have your skeletal muscle, you have your cardiac muscle, or cardiac myocyte, and you have your pacemaker cells, and of course you have your smooth muscles. But not just any smooth muscle. There is something called single unit smooth muscle, or unitary smooth muscle. They have gap junctions that connect them to each other. So if you stimulate one, the electric signal will be transmitted to the other cells. So these kinds of smooth muscles, which are known as single unit or unitary smooth muscles, are usually seen in the GIT, in the gut over there. Now, what we need to understand is we're going to talk about the action potential in the neuron, first of all. And it's quite different from that which is taking place in the cardiac muscle and pacemaker cells, and even some smooth muscles. They have different types of action potentials. But when we talk about action potential, we generally mean the type that's happening in the neuron or in the skeletal muscle. They're virtually the same. Now, the people who worked on action potential, your Hodgkin and Huxley, they actually worked on the neuron of a giant squid. So it's known as a loligo. Okay, that's known as a loligo. Or the giant squid. Now, why they used the giant squid is because it was a very, very large organism. It's quite massive. If you watched Pirates of the Caribbean, you would see that there was this creature called the kraken, which was the big squid that would just swallow ships. Yeah, so one of those giant squids was able to wash on to shore, and they were able to capture one of these and do the research on it. So they took the neurons of that squid because they were massive. They were quite big. The diameter of one of these axons was up to 0.5 millimeter to 1 millimeter in diameter. For that reason, people like to do research on action potentials and bioelectricity using the giant squid axon. Now, what they did was they put this giant squid axon in a setup where the neuron was able to survive. So they put it in a little bit of fluid, and the fluid mimicked the milieu interior, of that creature, and because of this we are able to sustain the neuron over here. Now in this diagram, which I'm focusing over here, I'm just showing you the axon and the setup over here, which allowed them to study the action potential and make our lives a little more difficult in physiology. But no, without action potential, there is no long distance rapid communication like this. It is one of the most efficient and most beautiful ways to allow thought processing signals from the brain to the muscle, to allow movement to take place, to your composition of music in your mind. All of these beautiful things happen because of electric activities happening in your neurons. And if we understand how this happens in the giant squid axon, we could understand how it happens in certain other tissues in the body. So we have this giant axon, and here you could see that there is something called a stimulator over here on the upper left hand corner of this diagram. So this stimulator, it gives a current. Now there are two types of currents. There is a catalectric current, and there's an anelectric current that this stimulator can make. When we try to stimulate this cell and create an electrical signal, in this case the action potential, we are going to use a catalectric current, and I'm going to emphasize on this in some more time and elaborate on this in some more time. So we have a neuron, we have this axon over here, and there is a stimulator on one side. On the far end, on the other side, in the right hand corner of this diagram, I have something over here called the cathode ray oscilloscope. Now the CRO, or cathode ray oscilloscope, it is meant to measure, it's actually a voltmeter, which measures the potential difference across the membrane over here, because I've put one of the electrodes outside, which you could see I'm just coloring in black over here, and one more on the inside over here. So it tells me the difference in the charge on the two sides of the membrane, what we call voltage, right? Or you could say the potential difference across the membrane, because what is potential difference? It happens when you separate charges. So when you separate charge across some sort of a barrier, then there is this potential for electricity to form, for a buildup of energy, and that's your potential difference in very simple terms. Forgive me, I'm not a physicist or anything, but I can just give it to you in simple terms. Alright, so what happens here is this axon has got some channels over here, ion channels, which allow the movement of certain ions. I've drawn red and green. The red is sodium, the potassium is in green. So sodium channels are specific. They allow sodium to come in when a certain voltage has been reached in the inside of the neuron. And same thing with potassium. It allows potassium to this one enter or leave the cell depending on what is the voltage across the membrane. So now let's just recap one more time that sodium tends to be more on the outside of the cell. So outside the cell, in a human being, for example, your sodium is around 140 milliequivalents per liter. In terms of potassium, it's around 4 milliequivalents per liter. And sodium in the axon is around 14 milliequivalents per liter. And potassium is around 140 milliequivalents per liter. So you need to know these values, because it tells you that when these channels are opened, because of the different voltages that voltage changes that take place, you'll be able to guess where these ions move. Now let's elaborate a little more over here. Before we go into the sequence of events, I'll just tell you how these sodium channels and potassium channels look, so you can understand what's going on. So this is a sodium channel. I'm just drawing a cell, hypothetical cell, and here I have the sodium channel that I'm drawing in red. Alright, so let's just say this is the ion channel, and there are two gates. Okay, this is the ECF, the outside ECF, and the inside is the intracellular fluid, ICF. Now, there are two gates over here. One is the M gate, M. This is M, and this gate is the H gate, H. They're named after two constants which Hodgkin and Huxley used in their formulas when they were trying to guess how the sodium channels worked using mathematical modeling. They did this without an electron microscope, and they were able to accurately guess that there must have been two gates based on their mathematical formulas. That was quite impressive. Now, the M gate, usually in a resting state, when the neuron is not firing and action potential, is closed, while the H gate is open. And this is how this cell is, how the sodium channel is in the resting state. So this is the resting state. And you could say that the sodium channel is cocked, like a gun ready to fire. Okay, it's resting. Okay, and now what happens is this, in this situation, when the neuron is resting and it's not firing, the inside of the cell is more negative than the outside. This is not because we have something called resting membrane potential, where in humans the RMP is minus 90 millivolts. But if you look in your books, you might sometimes see in this action potential setup that the RMP can be represented as minus 70 millivolts. This minus 70 is in the squid, in this giant squid, and minus 90 is in the human neuron. And most of our cells are around minus 90 when it comes to RMP. So let's just write neuron over here. So why is this difference over there? Well, because we're different species, so we have a little difference in our permeability in our channels, so don't think too much about it. Just remember that minus 70 represents the oligos and minus 90 is the humans. Now, this gate that you see, the M gate is closed and the H gate is open when the RMP is minus 90 millivolts, meaning the inside is a little more negative than the outside. Now, what happens is you get a situation where you've depolarized the cell. Depolarization means you're getting rid of this potential difference where the inside is more negative than the outside. That means you're making the cell more positive. Actually, depolarization applies to whenever you make the cell positive. Even if you make it too positive and you reverse the polarity, it doesn't matter. You made the cell positive, you depolarize. You make the cell negative, you repolarize. Now here we have a situation where we make the cell a little positive. However, you can make the cell positive by using an electrode, by stimulating the area, by building up positive charges here, or sometimes if it was a neuron, just think how it works. A neuron or any other tissue. Neurons communicate by releasing neurotransmitters, and these neurotransmitters can stimulate other neurons by opening channels, ion channels, that build up enough charge and cause a continuous chain reaction to take place. So that's how these cells work. Well, when you make the cell more positive on the inside now, bring in some positive charge one way or another, these voltage gated channels, which are sensitive to the voltage, will swing open. So I'm going to draw this channel again, and what happens is the M gate opens up and the H gate starts to close. Think of them as though they are being connected by an invisible rubber band over there. So the M gate swings open, it flings open, and the H gate suddenly starts to close over there. And this is when you just depolarize the cell quite a bit, and this allows the sodium to gush into the cell. Because why? If we remember, sodium is around 140 on the outside, and on the inside your sodium levels are 14 in the intracellular fluid. So don't forget this. Sodium tends to gush in, and this happens for a small time. The thing is, these sodium channels, they close up, and they close up in a different configuration, where the M gate is now open and the H gate is closed. over here. Now this is the inactive state. Okay, no more sodium is going to come in. There's a lot of sodium that just gushed in over there, and you're not going to get any more sodium in because this H gate has closed. There's a funny way of remembering this. The M gate is like the main gate, so it's to the outside, and the H gate it's like the beginning for the word home, so your house is inside the main gate. So you can always remember this as the H gate is on the inside and the M gate is on the outside. All right, so now this state which you see, this third state over here, this is the state where your sodium channels are not going to allow any more sodium in. What happens is there's something called time-dependent closure. Time-dependent closure. Meaning to say the thing closes in a matter of maybe a millisecond or two. So within 1 or 2 milliseconds, this gate closes. And how much sodium comes in? It comes in within that 1 or 2 milliseconds. And then you get this configuration. And there's no more positivity entering into the cell. So, and you can't get another action potential until you make this inactive, what has now become an inactive sodium channel, back to its original cocked state, its resting state over here, like we said in the beginning over here. So how do you make this inactive sodium channel active again? You got to repolarize the cell. You have to make the cell negative. So when you start making the cell negative after the depolarization, that is to say the inside of the cell starts to get more negative, closer to its RMP, it's trying to restore its resting membrane potential, then these channels start to flip back to this original resting configuration, where it is able to cause another action potential. So now if we look at how this works over here, you have your sodium channels. You have they're very important for the depolarization process. See, sodium gushes in when you activate enough number of sodium channels. And when it comes to potassium, it's not so hard to talk about that. I just want you to know that potassium channels have one single gate, and potassium, if you look at the level over here, potassium is around 140 milliequivalents in the cell, and potassium is just 4 milliequivalents outside the cell. Now you open up this potassium channel and potassium is going to leave the cell over here because it follows the concentration gradient. It goes along the concentration gradient and takes its positive charge along with it outside. Hence it makes this cell more negative, and this is what we call repolarization. So one more point to remember, potassium channels tend to be sluggish, meaning they tend to close themselves a little later than they should, and I'll explain as we go ahead over here. So what happens is we have a situation where we are able to initiate an electric impulse, a signal, with depolarization followed by repolarization because of these different ion channels, and that causes communication of signal and spreading of the signal around over here. So the sodium gushes in and spreads and opens more sodium channels and more sodium channels, and potassium channels open up because they also are exposed to the positivity in the cell, and potassium leaves the cell, making the cell negative. So let's just go through the steps over here of what happens. We have an axon, and this axon has got sodium channels, and I'm just drawing a few sodium channels here in red. Let's just take red for sodium, okay? And we have the potassium channels interspersed amongst them, because fluid mosaic model, you've got all of these interspersed around. And what happens is sodium can gush in when you activate it. So how is that activated? Remember, there was a stimulator over here. So there's a little stimulator over here which can develop a little current over here. So this is your stimulator. Now what does the stimulator do? It can make a catalytic current. get electric